As the night settles in and the world outside grows softer and quieter, let us begin a gentle journey into one of the most fundamental sciences of life, biochemistry. It is a word that may sound large and technical, perhaps even heavy, but here, under the calm of night, we will let it unfold slowly, peacefully, and without hurry. Think of it less as a complicated subject and more as a quiet story of how the smallest parts of life work together to keep everything alive. Biochemistry, in its simplest sense, is the study of life at the level of molecules. It is where chemistry meets biology, where atoms and bonds give rise to living systems, and where invisible reactions shape the beating of hearts, the growing of plants, the flight of birds, and even the thoughts passing gently through your own mind. Every breath you take, every cell that stirs in your body, is guided by the principles of biochemistry. It is as if an entire orchestra of molecules is playing in silence, creating the symphony of life. And while we cannot hear these instruments or see them directly, their presence is constant, subtle, and essential. At its core, biochemistry asks simple questions. How do living things take in energy, and how do they use it? How are nutrients broken down, and how are they rebuilt into the tissues of life? How do cells communicate, coordinate, and sustain balance in an ever-changing environment? The answers to these questions are not abstract. They are the quiet realities unfolding within you at this very moment. To understand biochemistry, we begin with the basic building blocks of matter. The smallest unit is the atom, tiny beyond comprehension, yet full of possibility. When atoms join together, they form molecules, and some of these molecules are particularly important for life. Water, for example, is one of the simplest molecules, yet without it, life as we know it could not exist. Its shape, with two hydrogen atoms joined to one oxygen, gives it properties of dissolving, transporting, and stabilizing qualities that make it the perfect medium for life's chemistry. Beyond water, life also depends on molecules that contain carbon. Carbon is special because it can form 4 bonds at once, creating chains, rings, and complex structures. From this flexibility, carbon builds sugars, lipids, proteins, and nucleic acids, the 4 great classes of biomolecules. Sugars provide energy, lipids form membranes and store fuel, nucleic acids carry genetic instructions, and proteins, as we will see, act as the versatile workers of the cell. Among proteins, there is one group with a particularly quiet yet powerful role: enzymes. Enzymes are the hidden caretakers of chemistry. They are proteins that make reactions possible, that allow processes to occur at the speed of life. Without them, the chemistry of a single breath, or the beating of your heart, would take far too long to sustain life. Enzymes do not force reactions. They guide them gently, lowering the barriers that stand in the way, so that molecules can meet, transform, and move forward. They are like soft-handed guardians, watching over every transformation in the body. But before we speak more of enzymes, we must return to a central theme of biochemistry: energy. Energy is the invisible currency of life. It cannot be seen directly, but we can observe its effects: the warmth of the body, the motion of muscles, the growth of cells. In physics, energy is the capacity to do work, to cause change, to move matter. In biology, it is the quiet flow that keeps systems alive. Imagine, for a moment, the food you eat. A piece of fruit, perhaps an apple, is more than just taste and texture. It is a collection of molecules rich in stored energy. That energy was first captured by the apple tree from sunlight, converted into sugars through photosynthesis, and packed into the flesh of the fruit. When you eat the apple, your body breaks those sugars down, releasing the energy stored within their bonds. That energy then fuels your cells, allowing them to carry out their quiet, endless tasks. This cycle, from sunlight to sugars to cellular energy, is the essence of life's flow. Biochemistry is the map that helps us understand it. It is a study of how molecules are transformed, how energy is stored, and how balance is maintained. And at the heart of this map, always, are enzymes. In the hours ahead, we will move through this landscape carefully. We will learn how energy is defined in living systems, how it is captured and released, and how enzymes act as the catalysts that make every reaction possible. We will explore the great cycles of life, the way plants gather energy from the sun, the way animals release energy from food, and the way all organisms rely on delicate molecular choreography to sustain themselves. But for now, in this gentle introduction, it is enough to remember a few simple truths. Life is chemistry, but not ordinary chemistry. It is chemistry, guided by enzymes, powered by energy, and sustained by balance. Every moment of your existence is a dance of molecules, a harmony of transformations, and a silent flow of energy from one form into another. To think of biochemistry in this way is not to be overwhelmed, but to be comforted. It is a reminder that life is not random chaos, but a carefully maintained system of order and flow. Even in the smallest cell, there is direction, coordination, and purpose. And as you rest, your body continues this work effortlessly, with enzymes guiding, with energy flowing, and with balance maintained. Thus, as we begin, let us hold this image: the quiet world within, where molecules meet and part, where energy flows like a river, and where enzymes stand as timeless guides, ensuring that life continues, gently and steadily, in every moment. 2. The nature of life's energy. Energy is a word that we use in many different ways. We speak of having energy when we feel awake and lively. We say we are low on energy when tired. In the physical sciences, energy is something more precise. It is the capacity to do work, the ability to cause change, or to move matter from one state into another. In the quiet realm of biochemistry, energy is everywhere. It is the invisible current that flows through living systems, a constant exchange from one form to another, a leaf turning sunlight into sugar, a bird taking flight, or even a single cell copying its DNA. Each is possible only because energy moves, transforms, and is used carefully. Without this flow, life would be still. Let us imagine energy as a soft river running through the living world. Sometimes it rushes quickly, as in a heartbeat or a muscle contraction. Sometimes it flows slowly, stored quietly until needed. But always, it moves, never truly disappearing, only shifting from one form to another. Scientists describe energy in several ways. There is kinetic energy, the energy of motion. It is found in the beating of wings, the vibration of molecules, the movement of ions across a membrane. There is also potential energy, energy stored and waiting, like water behind a dam or a molecule holding chemical bonds. In living systems, it is often this chemical potential energy that matters most, for it can be released and transformed into movement, heat, or growth. Chemical bonds are tiny stores of potential energy. When atoms share or exchange electrons to form a bond, they settle into arrangements that hold energy within them. Some bonds are easy to rearrange, releasing energy readily, while others are more stable, holding on to their energy tightly. Living organisms depend on the ability to access, release, and redirect this chemical energy in a controlled way. Consider a simple sugar, such as glucose. Its carbon, hydrogen, and oxygen atoms are arranged in a ring that contains a rich store of energy. When enzymes in your cells break the bonds of glucose, they release this energy step by step. The energy does not simply vanish. It is harnessed to fuel reactions, pump ions, and maintain the delicate state of life. What once was stored as potential energy in a molecule becomes useful energy in a cell. At the same time, energy is never created from nothing. This is one of the most fundamental laws of physics, called the conservation of energy. Energy may change form, but it cannot appear from nowhere, nor can it vanish into nothing. For living beings, this means that every movement, every thought, every breath is paid for by the transformation of energy taken from the environment. For plants, the source of this energy is sunlight. Through photosynthesis, they capture the energy of photons streaming from the sun, transforming light into the chemical bonds of sugar. For animals, including humans, the energy comes from food. Plants provide sugars, starches, and fats. Animals provide proteins and additional stores of fuel. By eating, we take in molecules already rich in stored chemical energy. Through digestion and cellular respiration, we release that energy and make it usable. A central feature of biological energy is that it is managed in small, careful steps. A log burning in a fire releases all its energy at once. In heat and light, a cell cannot afford such violence. It must extract energy gently, using enzymes to guide reactions slowly, in pathways that capture and conserve as much as possible. Instead of a sudden blaze, the cell relies on a controlled series of small transformations, each releasing a portion of energy, each feeding into the next. The cell's main energy carrier is a molecule called ATP adenosine triphosphate. It is sometimes called the energy currency of the cell because it is used in almost every process. Its three phosphate groups are linked by bonds that hold potential energy. When one phosphate is removed, ATP becomes ADP, releasing usable energy that can power a reaction. Later, with the help of enzymes, ADP can be recharged, gaining another phosphate and once again becoming ATP. This cycle repeats endlessly, a molecular heartbeat within every living cell. It is important to understand that energy is not just a fuel, it is also a measure of order. Living systems must constantly fight against entropy, the natural tendency toward disorder. Left alone, molecules drift into randomness. Energy allows life to maintain structure, to build order, to resist decay. A cell uses energy to repair itself, to reproduce, to keep its interior conditions stable despite changes outside. Without energy, life quickly fades into stillness. We can also think of energy as a bridge between the living and non-living worlds. Rocks and rivers have energy, but they do not use it. In organisms, energy becomes purposeful. A falling stone is moved by gravity, but a climbing vine uses energy to reach upward toward the light. The difference lies in the ability of life to capture energy, to store it, and to direct it toward survival. Throughout the earth, this flow of energy links all living things. The sun's energy enters plants, which store it in sugars. Animals eat the plants, releasing the stored energy for their own movements and growth. Other animals eat them in turn. When organisms die, decomposers break down their remains, returning molecules to the soil and atmosphere, ready to be used again. The cycle continues, a vast and quiet circulation of energy that binds all life together. Even within your body, this circulation is continuous. Each heartbeat depends on a rush of ions across cell membranes, powered by ATP. Each breath requires muscles contracting, driven by chemical energy. Even as you rest, countless enzymes are working to transform energy into the small actions that keep you alive. Repairing proteins, copying DNA, sending signals along nerves. Energy is the quiet force that sustains life's rhythm. Thus, when we speak of energy in biochemistry, we speak not of abstract numbers, but of the most intimate truth of existence. To live is to transform energy, to take it in, to use it, and to pass it along. Every organism, from the smallest bacterium to the tallest tree, participates in this flow. And while the details may seem complex, the essence is simple. Life is energy in motion. As we move forward, we will look more closely at ATP, the special molecule that carries energy within cells, and at enzymes, the remarkable proteins that make energy transformations possible. For now, let us rest in the understanding that energy, though invisible, is the gentle current running through every moment of life. 3. ATP, the cell's energy currency. When we look closely at life, down to its smallest functioning parts, we discover that living systems have chosen a remarkably elegant way of handling energy. The molecule that carries out this work, serving as a kind of universal coin or currency, is adenosine triphosphate, often simply called ATP. ATP is not a rare molecule. In fact, it is one of the most abundant substances inside every living cell. At this very moment, trillions of ATP molecules are at work within your own body. They are constantly being used, broken down, and reassembled, flowing endlessly in cycles. Even as you rest in stillness, ATP molecules are quietly fueling every heartbeat, every blink, and every subtle chemical adjustment needed to maintain balance. To understand why ATP is so important, we must begin with its structure. The name itself gives clues. Adenosine refers to the base adenine, joined to a sugar called ribose. This forms the core of the molecule. To this core are attached 3 phosphate groups, hence the triphosphate. These 3 phosphates are linked in a chain, one after another, extending from the ribose. It is in this phosphate chain that ATP holds its power. The bonds between these phosphate groups are high in potential energy. They are sometimes called high-energy bonds, not because they contain more energy than other bonds, but because breaking them releases energy in a way that cells can easily harness. When the final phosphate group is removed, ATP becomes ADP, adenosine diphosphate, and a burst of usable energy is released. This reaction, the hydrolysis of ATP, is one of the most common events in all of life's chemistry. The cell pairs this release of energy with countless tasks: contracting a muscle fiber, transporting molecules across a membrane, or attaching a new building block during the synthesis of proteins. Each time, ATP serves as the provider, giving just enough energy for the job. What makes ATP especially remarkable is its reversibility. Once ATP loses a phosphate to become ADP, it is not discarded. Enzymes within the cell can add a new phosphate, restoring ATP and allowing it to be used again. This cycle, ATP to ADP and back again, turns endlessly, like a rechargeable battery being used, recharged, and used once more. In fact, the turnover is astonishing. A single human body may recycle the equivalent of its entire weight in ATP molecules each day. You are not carrying a large store of ATP like coins in a jar. Rather, your cells are constantly minting new molecules, using energy from food or sunlight, and spending them instantly on the work of life. ATP powers a vast range of processes. At the molecular level, it is used to drive what are called endergonic reactions, reactions that would not occur spontaneously because they require an input of energy. By coupling them to the hydrolysis of ATP, the cell makes these reactions possible. For example, building complex molecules like proteins or DNA strands requires energy. ATP provides it, ensuring that growth and repair can occur. Another vital use of ATP lies in movement. Inside cells, proteins called motor proteins use ATP to change shape, to crawl along fibers, and to carry cargo. These tiny movements, multiplied countless times, are what make muscle contraction possible, what allow nerve cells to transport signals, and what keep the intricate machinery of life in motion. ATP also powers transport across membranes. Cells are not passive bags of molecules. They are carefully regulated spaces, full of concentration gradients and delicate balances. Often, substances must be moved against these gradients, pushed into areas where they would not naturally flow. This active transport requires energy, and once again, ATP is the molecule that pays the cost. Pumps in the cell membrane bind ATP, use its energy to change shape, and move ions or nutrients into place. Even signaling in the body often involves ATP. In many cases, ATP itself acts as a signaling molecule, carrying messages between cells. It can also be used to attach phosphate groups to proteins, a process called phosphorylation. This small modification acts as a switch, turning enzymes or other proteins on or off, and thereby controlling vast networks of cellular activity. It is no exaggeration to say that ATP is the heartbeat of biochemistry. Without it, none of the intricate processes of life could continue. But it is important to remember that ATP does not appear on its own. It must be produced, and the pathways of its production are among the most studied and most fascinating in biology. In animals and many other organisms, ATP is generated largely through cellular respiration, a process that begins with the breakdown of glucose. Enzymes guide this breakdown in careful steps, capturing energy little by little and storing much of it in the bonds of ATP. In plants, ATP is produced as well during photosynthesis, using light to drive the addition of phosphate groups. No matter the source, sunlight, or food, the result is the same: a steady flow of ATP to power life. ATP is sometimes compared to money in an economy. Food molecules like glucose or fat are the stored wealth, large and rich but not directly spendable. ATP is like cash, small, immediate, and usable in every corner of the system. Just as money is exchanged for goods and services, ATP is exchanged for movement, for synthesis, for transport. Without currency, an economy stalls. Without ATP, life itself comes to a halt. And yet, despite its centrality, ATP is a simple molecule. Its strength lies not in complexity, but in universality. From the simplest bacteria to the most complex animals, ATP is the chosen carrier of energy. This unity is a reminder of life's shared heritage, stretching back billions of years to the earliest cells that first harnessed ATP's power. As you listen now, your body is a quiet landscape of ATP transactions. In every cell, Phosphates are being clipped away and reattached, releasing energy, then restoring it again. Each cycle is measured, precise, and vital. This constant rhythm never stops while you live. It continues through waking and sleeping, through thought and silence, an endless exchange of energy in molecular form. Thus, when we speak of ATP as the currency of life, we mean not only its usefulness, but its universality. It is the common denominator of biology, the quiet coin that all living things accept. In the stillness of the night, it is comforting to know that within you, this circulation of energy continues, steady and unfaltering, keeping you alive with every passing moment. 4. Enzymes as biological catalysts. Now that we have come to know ATP as the gentle coin of energy within living cells, let us turn our attention to the remarkable helpers that make its constant circulation possible. Enzymes. Without enzymes, the energy stored in ATP, glucose, or any nutrient would remain locked away, unavailable at the speed life requires. With enzymes, however, reactions unfold in perfect rhythm, fast enough to sustain life, yet subtle enough to be controlled. Enzymes are proteins, folded chains of amino acids that twist into intricate shapes. At first glance, a protein might appear as nothing more than a cluster of atoms. Yet within this structure lies an extraordinary talent. Enzymes act as catalysts. A catalyst is a substance that speeds up a chemical reaction without being consumed by it. In essence, it makes a reaction easier, lowering the barrier that stands in the way. This barrier is known as activation energy. Every chemical reaction, whether in the air, in water, or within a living cell, requires a certain input of energy before it can proceed. Imagine a boulder resting in a valley. To roll into the next valley, it must first be pushed up and over a hill. That hill is the activation energy. The boulder already holds potential energy, but without help, it will not cross the ridge. In the quiet world of biochemistry, enzymes act like gentle guides, lowering the height of the hill. They do not push the boulder with brute force. They simply reshape the path, making it easier to cross. With the barrier lowered, the reaction proceeds quickly, smoothly, and continuously. This is why enzymes are essential. Without them, the reactions of life would still happen, but so slowly that life itself would not be possible. For example, the breakdown of hydrogen peroxide into water and oxygen occurs naturally, but at an imperceptible pace. In your cells, the enzyme catalase performs this task in fractions of a second, preventing harmful buildup and maintaining balance. Each enzyme is highly specific. It recognizes only certain molecules, known as substrates. The part of the enzyme that interacts with its substrate is called the active site. This site is shaped in a way that complements the substrate, much like a lock is designed to fit a particular key. When the substrate enters, it binds gently to the enzyme, and the reaction takes place. There are two main ways of describing this interaction. The first is the lock and key model, which imagines the enzyme as a rigid structure, perfectly fitted to its substrate. The second is the induced fit model, which suggests that the enzyme is more flexible, adjusting its shape slightly to embrace the substrate more closely. In either case, the result is the same. The enzyme stabilizes the reaction, lowers the activation energy, and allows the transformation to occur. Consider, for example, the enzymes involved in digestion. Amylase, found in saliva, breaks starch into simple sugars. Proteases break proteins into amino acids. Lipases break fats into glycerol and fatty acids. Each enzyme is tuned to its particular substrate, and together they transform food into usable molecules. Without enzymes, the nutrients in food would remain locked away. With them, digestion flows effortlessly, fueling life. But enzymes do not only act in digestion, they participate in every process within the cell. Some build molecules, others break them down. Some rearrange atoms within a compound, while others transfer groups of atoms from one molecule to another. Enzymes are involved in copying DNA, repairing damaged molecules, sending signals across membranes, and producing ATP. In fact, nearly every reaction in your body depends on at least one enzyme. What makes enzymes especially fascinating is their efficiency. A single enzyme molecule can process thousands of substrate molecules per second. And yet, when the reaction is complete, the enzyme remains unchanged, ready to act again. It is a quiet worker, tireless and precise. Despite their power, enzymes are delicate. Their structure depends on weak interactions between atoms, and these can be disrupted by changes in temperature, pH, or chemical environment. If the conditions shift too far, the enzyme may lose its shape, a process known as denaturation. A denatured enzyme can no longer bind its substrate, and the reaction comes to a halt. This sensitivity is why living organisms must carefully regulate their internal conditions, keeping temperature, acidity, and other factors within narrow ranges. Enzymes also operate under control. Cells do not allow reactions to proceed unchecked. Instead, they use regulators molecules that can activate or inhibit enzymes as needed. Some regulators bind directly to the active site, blocking the substrate. Others attach elsewhere on the enzyme, subtly changing its shape and altering its activity. These controls ensure that energy is not wasted, that molecules are produced only when required, and that balance homeostasis is maintained. There are even feedback mechanisms, where the product of a pathway signals back to shut down the very enzymes that created it. Imagine a factory that produces a substance until the shelves are full. Once the supply is sufficient, the machines slow down or stop. In the cell, enzymes behave in the same way. When ATP levels are high, the enzymes that produce ATP slow down. When levels fall, the enzymes are released from inhibition, and production resumes. All of this, the specificity, the efficiency, the regulation makes enzymes not just catalysts, but guardians of life's chemistry. They are the reason glucose can be broken down step by step, rather than all at once in a fiery release. They are the reason DNA can be copied with precision. They are the reason your muscles contract, your nerves fire, and your heart beats. And yet, enzymes themselves are made from the same building blocks as all proteins. Chains of amino acids folded into unique three-dimensional shapes. It is their shape that grants them their activity. This simplicity is humbling, a reminder that from basic principles, life has crafted remarkable solutions. In the natural world, enzymes extend beyond the body. In soil, they help break down organic matter, recycling nutrients for plants. In oceans, they assist in the cycling of carbon and nitrogen. In technology, humans have learned to harness enzymes for many purposes, from brewing beer to making cheese, from cleaning laundry to producing medicines. Though invisible, enzymes shape much of the world we experience. As you rest tonight, enzymes are busy within you, working silently. They are splitting ATP, joining phosphates, repairing DNA, digesting nutrients, and producing the molecules you will need tomorrow. They do so without pause, without error, and without fanfare. You may never feel their presence directly, but every moment of your life depends on their gentle labor. Thus, when we think of enzymes, we can imagine them as quiet custodians, tending to the endless fires of life, ensuring that energy flows smoothly, that balance is kept, and that life continues. Without them, time within the body would slow to a crawl. With them, life moves in harmony, every reaction perfectly timed, every process carefully guided. 5. The structure of enzymes. To understand enzymes more deeply, we must turn our attention to their form, the quiet elegance of their structure. They are not simple strings of atoms, but folded, coiled, and shaped into three-dimensional forms of remarkable precision. It is this structure, above all else, that gives them their power. At the simplest level, enzymes begin as chains of amino acids. These amino acids are linked together by peptide bonds, one after another, like beads on a thread. There are 20 different types of amino acids, each with its own chemical personality. Some are small and flexible, others bulky and rigid. Some carry charges, while others prefer to hide from water. When a cell builds a protein, it selects the sequence of amino acids carefully, following the instructions written in DNA. This sequence is the primary structure of the protein, the basic order of its amino acids. But this is only the beginning. Once the chain is formed, the amino acids begin to interact with one another. Hydrogen bonds form between nearby regions, causing the chain to fold into coils and sheets. These patterns are called the secondary structures alpha helices, which look like spirals, and beta sheets, which look like folded ribbons. From there, the protein folds further. Interactions between distant parts of the chain cause it to twist, bend, and collapse into a compact shape. This is known as the tertiary structure, the full three-dimensional form of the protein. For enzymes, it is within this folded structure that the active site emerges, the pocket or groove where the substrate will bind. Some enzymes are even more complex, consisting of multiple chains, called subunits, which join together to form a quaternary structure. Hemoglobin, for example, though not an enzyme, is a protein with 4 subunits working together. Enzymes with quaternary structures gain additional abilities, such as cooperative binding, where one subunit influences the activity of others. It is within the folds, grooves, and surfaces of these structures that the magic of enzymes occurs. The active site is shaped to complement the substrate, ensuring specificity. A single change in the amino acid sequence can alter the shape, sometimes eliminating activity altogether. This is why mutations in DNA, which change amino acids, can have such profound effects on life. The structure must be precise for the enzyme to function. The active site itself is not a passive pocket. It is lined with amino acids whose side chains interact with the substrate. Some attract it with charges, others form hydrogen bonds, while still others push water molecules aside to create a dry space for the reaction. The enzyme may bend the substrate slightly, holding it in a strained position that makes it easier to react. In this way, the enzyme does not merely wait for the reaction, it actively prepares the molecules, guiding them toward transformation. Let us consider an example. The enzyme hexokinase, one of the first steps in glucose metabolism, binds to a glucose molecule and an ATP molecule at the same time. Within its active site, it holds the glucose and the ATP in just the right orientation so that a phosphate group can be transferred. Without the enzyme, the two molecules might never collide correctly. With hexokinase, the reaction occurs smoothly, transferring energy from ATP into glucose, beginning the pathway of respiration. This example reveals another truth. Enzymes do not add energy to reactions. They do not change the starting materials or the final products. What they do is lower the activation energy, making the path easier, the hill gentler to climb, and this is only possible because of their structure, because of the way their folds and shapes cradle the substrate and coax it along. Enzyme structures are not rigid. Though we may imagine them as locks and substrates as keys, the reality is more fluid. In the induced fit model, the enzyme shifts slightly when the substrate binds. This flexibility allows the enzyme to grip the substrate more tightly, aligning atoms precisely for the reaction. It is like a handshake, where both hands adjust to meet comfortably. Some enzymes also rely on helpers, known as cofactors or coenzymes. These may be metal ions, such as zinc or magnesium, or organic molecules, such as vitamins. These helpers sit within the enzyme structure, providing chemical abilities that amino acids alone cannot. For example, the enzyme carbonic anhydrase, which helps balance carbon dioxide and bicarbonate in the blood, relies on a zinc ion at its core. Without the zinc, the enzyme would not function. The structure of enzymes can also include multiple domains, regions of the protein with distinct functions. One domain might bind the substrate, another might interact with regulatory molecules, and another might anchor the enzyme to a membrane. Together, these domains allow the enzyme to operate as part of larger networks, integrated with the needs of the cell. Despite their strength, enzyme structures are fragile. Heat can cause the weak bonds that hold the protein in place to break, unfolding the shape and destroying activity. Changes in acidity can alter charges, disrupting interactions within the protein. When this happens, the enzyme denatures, losing its form and its function. This sensitivity is why life depends so strongly on homeostasis, the maintenance of stable internal conditions. A small change in temperature or pH can silence enzymes, halting the processes of life. And yet, life has also found ways to adapt enzymes to extraordinary environments. Bacteria that live in boiling springs have enzymes that remain stable at high temperatures. Others, dwelling in icy oceans, have enzymes that remain flexible and active at near-freezing conditions. These variations remind us that while structure is delicate, it can evolve, adjusting to different worlds. When we look at enzymes through the lens of structure, we see not just proteins, but sculptures of life. They are crafted with precision, each fold and twist shaped by billions of years of evolution. Their beauty lies not only in form, but in function. Each groove and pocket serving a purpose, each movement guiding a reaction. As you rest now, the enzymes within you are folded into their perfect shapes, cradling substrates, guiding reactions, and sustaining life. Their structures, though invisible, are as real and intricate as any cathedral or mountain range. They are quiet architectures of existence, built not of stone or earth, but of atoms, woven into patterns that hold the power of life itself. 6. Energy pathways in cells. Now that we have met enzymes and seen how their structure enables their work, let us step back and watch them in action, not in single reactions, but in entire pathways. For life does not depend on isolated chemical events. Instead, it relies on sequences, chains of reactions, each one guided by an enzyme, and each step feeding smoothly into the next. These are the energy pathways of the cell, the quiet routes by which energy flows. An energy pathway is like a winding river. It begins at one source, travels through many bends and turns, and eventually empties into a larger body of energy that can be used by the cell. Along the way, enzymes stand as gatekeepers at every bend, ensuring that the water flows steadily, not too fast, not too slow. If even one gatekeeper were absent, the river might become blocked or spill into an unintended direction. This is why pathways must be complete and continuous, every enzyme in its place, every step accounted for. One of the most central energy pathways is cellular respiration. This is the process by which glucose, the simple sugar found in fruits, grains, and countless other foods, is broken down into usable energy. Glucose carries within it chemical bonds that store potential energy. The task of the cell is to release that energy carefully, without waste, and to capture it in the form of ATP. Cellular respiration occurs in stages, like a long journey divided into chapters. The first chapter is glycolysis. This word means sugar splitting, and that is exactly what happens. A molecule of glucose, with its 6 carbons, is split into 2 molecules of pyruvate, each with 3 carbons. This pathway occurs in the cytoplasm, the watery interior of the cell, and it does not require oxygen. Along the way, a small amount of ATP is produced, and electrons are captured by a carrier molecule called NADH. From glycolysis, the journey continues. If oxygen is available, the pyruvate enters the mitochondria, the tiny powerhouses of the cell. Here, in a process known as the citric acid cycle, or Krebs cycle, the carbons of pyruvate are released as carbon dioxide. With each turn of the cycle, more electrons are captured by NADH and another carrier. Only a small amount of ATP is produced directly in this cycle, but the true purpose is to harvest high-energy electrons, which will be used in the final stage. The last chapter is the electron transport chain, found in the inner membrane of the mitochondrion. Here, enzymes and protein complexes pass electrons from one carrier to the next, like a line of workers passing a bucket down a row. At each step, energy is released, and this energy is used to pump protons across the membrane, creating a gradient, a store of potential energy, like water behind a dam. Finally, the protons flow back across the membrane through an extraordinary enzyme called ATP synthase. This enzyme uses the flow of protons, much like a water wheel uses flowing water, to generate large amounts of ATP. The oxygen we breathe serves as the final acceptor of electrons at the end of the chain, combining with them and with protons to form water. This is why oxygen is essential for aerobic respiration. It is the quiet sink that keeps the flow moving. Through this series, glycolysis, the citric acid cycle, and the electron transport chain, the cell transforms the energy of glucose into ATP. From a single molecule of glucose, up to 30 or more molecules of ATP may be produced. Each one of these ATP molecules is then spent on the countless tasks of life. Not all organisms follow the same path. Some live in environments without oxygen, and they rely instead on fermentation. In this case, glycolysis still occurs, but the pyruvate is converted into other products, such as lactic acid or ethanol, to regenerate the carriers needed for the process to continue. Fermentation produces far less ATP than aerobic respiration, but it is enough for certain cells, and it allows life to persist even in the absence of oxygen. Energy pathways are not limited to glucose. Fats and proteins, too, can be broken down to provide fuel. Fatty acids enter the mitochondria and are converted into molecules that feed directly into the citric acid cycle. Proteins, after being broken into amino acids, can also be transformed into intermediates of the same pathways. In this way, the cell has flexibility. It can draw from many sources, always finding a way to sustain its energy needs. Pathways are not only about breaking molecules down, there are also anabolic pathways, which build complex molecules from smaller ones. For example, the synthesis of proteins from amino acids, or of DNA from nucleotides, are carefully guided by enzymes. These processes require energy, and so they are coupled to ATP hydrolysis, ensuring that growth and repair proceed smoothly. What makes these pathways truly remarkable is their interconnection. Glycolysis does not stand alone, nor does the citric acid cycle. They are woven into a larger web of metabolism, where the products of one pathway feed into another. If glucose is scarce, the body may turn to fats. If amino acids are abundant, they may be redirected into energy production. This flexibility ensures survival, allowing organisms to adapt to changing environments. Enzymes are the conductors of this metabolic orchestra. At certain key steps, enzymes act as checkpoints, their activity regulated by the energy state of the cell. If ATP levels are high, some enzymes slow down, reducing the flow. If ATP is low, the enzymes speed up, accelerating production. This feedback ensures balance. Enough energy to sustain life, but not so much that resources are wasted. The pathways of energy are therefore not chaotic, but ordered, balanced, and self-regulating. Each molecule has its place, each reaction, its timing, and each enzyme, its role. Together, they form a living network, a flow of energy that never ceases while life persists. As you rest tonight, these pathways are unfolding within you. Glucose from your meals is being split, electrons are being carried, protons are flowing across mitochondrial membranes, and ATP is being generated in countless numbers. All of this occurs silently, without effort on your part, sustained by enzymes that never tire, and by pathways that have been shaped by billions of years of evolution. Thus, when we speak of energy pathways in cells, we are speaking of life's most fundamental rhythm, the transformation of matter into energy, guided by enzymes, balanced by feedback, and sustained by the flow of ATP. It is a rhythm as constant as the beating of your heart, as quiet as the breathing of air, and as steady as the turning of the earth. 7. Enzymes in metabolic networks. Up to now, we have seen how enzymes guide individual reactions, and how those reactions connect into pathways such as glycolysis and respiration. But in truth, the cell is not made of separate, isolated chains of reactions. It is more like a great woven fabric, where each thread crosses others, and where enzymes tie everything together into a living whole. This interconnected web is what we call metabolic networks. Metabolism itself means simply change. In living organisms, it refers to all the chemical reactions that occur to maintain life. These reactions fall into two broad categories: catabolism, the breaking down of molecules to release energy, and anabolism, the building up of molecules to create structure and store energy. Together, catabolism and anabolism form a balance, one side tearing down, the other side rebuilding. Enzymes are the workers on both sides, guiding each reaction in harmony with the needs of the cell. Imagine, for a moment, a quiet city. Streets run in every direction, some leading to factories, others to markets, others to homes. Goods flow along these streets, being broken down, rebuilt, and distributed. Some roads are busy, carrying many travelers. Others are small side streets, only used at certain times. Enzymes are like the workers directing traffic at intersections, ensuring that the flow continues smoothly, preventing blockages, and rerouting when necessary. In the metabolic network, glucose is often the main traveler. If energy is needed, enzymes send it through glycolysis and respiration, generating ATP. But glucose can also be redirected into other pathways. It may be used to build nucleotides for DNA, or to create lipids for membranes, or even stored as glycogen for later use. The choice depends on signals within the cell, and enzymes at key points serve as decision makers, opening or closing routes as required. One such decision point occurs at the molecule pyruvate, the product of glycolysis. Enzymes can guide pyruvate into the mitochondria, where it enters the citric acid cycle for energy production. Or, in the absence of oxygen, other enzymes may redirect pyruvate into fermentation pathways, producing lactic acid or ethanol. The cell listens to its environment, and enzymes act accordingly, ensuring survival under changing conditions. The same flexibility is seen with amino acids, the building blocks of proteins. When food is abundant, enzymes direct amino acids into protein synthesis, supporting growth and repair. But in times of scarcity, other enzymes can break down proteins, sending amino acids into pathways that generate energy. Nothing is wasted. Molecules are constantly repurposed, depending on the balance of needs. This adaptability is possible because enzymes are not fixed in activity. They are regulated. Some are turned on or off by signals such as ATP, ADP, or NADH, the molecules that reflect the cell's energy state. If ATP is abundant, enzymes in catabolic pathways slow down, conserving resources. If ATP is low, those same enzymes speed up, pulling molecules through the pathways to restore balance. In this way, enzymes form feedback loops, ensuring that energy supply meets energy demand. Other enzymes are regulated by hormones, the signals that travel between cells and organs. In humans, for example, the hormone insulin encourages enzymes to store glucose as glycogen, while glucagon activates enzymes that release glucose back into the blood. Here again, enzymes respond to larger networks, linking the metabolism of individual cells to the needs of the entire body. Metabolic networks also extend outward, connecting organisms to one another. In a forest, the carbon fixed by photosynthetic enzymes in plants becomes food for animals. In animals, enzymes break the molecules apart, releasing energy for movement and growth. When organisms die, decomposer enzymes in fungi and bacteria recycle their molecules back into the soil, where plants can use them again. Across the biosphere, enzymes create a vast web of transformation, binding all life together through cycles of matter and energy. Another beautiful aspect of metabolic networks is their efficiency. Rarely does a pathway end with waste. Instead, the byproducts of one reaction become the starting materials for another. Carbon dioxide, released from respiration, becomes the raw material for photosynthesis in plants. Ammonia, produced by the breakdown of proteins, is used by certain bacteria to make nitrates for soil. Even lactic acid, produced during intense muscle activity, is carried to the liver, where enzymes convert it back into usable forms. At the heart of this efficiency is the principle of compartmentalization. In eukaryotic cells, different pathways occur in different organelles. The nucleus handles DNA, the mitochondria handle respiration, The chloroplasts handle photosynthesis, and so on. By dividing tasks into compartments, the cell prevents confusion and allows enzymes to work in specialized environments. It is as if the city of metabolism has districts, one for industry, one for storage, one for communication, each with its own skilled workers. Yet, even with compartments, the network remains unified. Molecules pass between organelles, carried by transporters, and guided by enzymes. Information flows as well, in the form of signaling molecules that adjust enzyme activity across the cell. The result is a dynamic, responsive system, always shifting, always adapting, yet always balanced. To grasp the scale of this, consider that a single human cell contains thousands of enzymes, each participating in dozens of pathways. Together, they perform millions of reactions every second, and yet, the cell is not overwhelmed. It is ordered, structured, and efficient because the network is regulated at every step. One of the great achievements of modern science has been mapping these networks. Diagrams of metabolism look like vast webs, with lines and arrows connecting countless molecules. To the untrained eye, they may appear overwhelming, but to biochemists, they reveal the elegance of life's design. No molecule alone, but all interwoven, all sustained by the tireless work of enzymes. As you rest, the glucose from your last meal may be flowing through glycolysis. Fatty acids may be feeding into your mitochondria. Amino acids may be diverted into protein synthesis or energy production. Every pathway is active, but none in isolation. Each one connects to others, forming a seamless web of transformation. Enzymes, in turn, are the silent weavers of this web. They choose routes, open gates, slow traffic, or speed it up. They do so not with thought or will, but through their shapes and interactions, guided by the subtle language of chemistry. Without them, the network would collapse into disorder. With them, metabolism becomes a quiet masterpiece of coordination. Thus, when we speak of enzymes and metabolic networks, we are speaking of life itself not as scattered reactions, but as a flowing, integrated whole. It is a reminder that even at the smallest scale, living things are not isolated parts, but connected systems, woven together by countless threads of energy and matter, all moving in balance, all guided by enzymes. 8 factors that influence enzyme activity. Enzymes, as we have seen, are the quiet workers of life's chemistry, shaping pathways, guiding energy, and weaving together the networks of metabolism. Yet, for all their power, enzymes are also delicate. Their activity depends on the conditions around them. Just as a musical instrument must be tuned to play harmoniously, enzymes must exist within the right environment to function properly. If those conditions shift too far, the gentle balance of reactions begins to falter. The first, and perhaps most important factor, is temperature. Enzymes are proteins, and their function depends on their three-dimensional shape. Within this shape are countless bonds, hydrogen bonds, ionic interactions, and subtle attractions that hold the folds in place. At moderate temperatures, these bonds are stable, and the enzyme remains active. As temperature rises, however, the bonds begin to vibrate more strongly. If the heat becomes too great, the bonds can break, and the enzyme unfolds, losing its shape. This process is called denaturation, and once it happens, the enzyme can no longer function. Think of it like a snowflake. At just the right temperature, a snowflake is a delicate, beautiful structure. But as warmth creeps in, the pattern collapses into liquid water. In much the same way, an enzyme loses its intricate folds if it becomes too warm. Life has therefore evolved to thrive at specific temperature ranges, ensuring that enzymes remain intact. For humans and many animals, enzymes work best near body temperature, around 37 degrees Celsius. At lower temperatures, the bonds hold firmly, but the molecules move more slowly. Enzymes still function, but their reactions take place at a reduced pace, like a clock ticking more slowly in the cold. This is why refrigeration slows the growth of bacteria. Their enzymes simply cannot keep up. At higher temperatures, the opposite occurs. Reactions speed up, but risk denaturation if the heat grows excessive. Life exists within this narrow window, a reminder of its fragility. In other environments, enzymes are adapted to different conditions. In hot springs, where water boils with volcanic heat, microorganisms known as thermophiles live with enzymes that remain stable at temperatures that would destroy most life. In icy seas, by contrast, enzymes and psychrophiles remain flexible even in near-freezing waters, allowing reactions to continue. These adaptations remind us of the diversity of enzyme forms, each finely tuned to the world in which it exists. Another crucial factor is pH, the measure of acidity or alkalinity. Every enzyme has an optimal pH, a range within which its structure and function are stable. Outside this range, charges on the amino acids may change, altering the shape of the active site or disrupting the bonds that hold the protein together. For example, enzymes in the human stomach, such as pepsin, are adapted to work best in highly acidic conditions, where the pH is around 2. In contrast, enzymes in the small intestine prefer a near-neutral environment with a pH closer to 7. Each is perfectly suited to its surroundings, and each would fail if moved to the other's home. The effect of pH is subtle but powerful. If acidity rises too far, enzymes may denature, just as they do with heat. If alkalinity increases, the same can occur. Thus, organisms carefully regulate their internal pH, ensuring that enzymes are never far from their optimal conditions. In humans, the blood maintains a nearly constant pH of 7.4. Even small shifts away from this value can be dangerous, because enzymes in critical pathways begin to lose efficiency. A third factor is substrate concentration, the availability of the molecules upon which enzymes act. When substrate is scarce, enzymes remain idle, waiting for molecules to arrive. As substrate concentration increases, the rate of reaction rises, for more active sites are filled, and more reactions proceed. But there comes a point of saturation, when every enzyme is already occupied. Beyond this point, adding more substrate does not increase the rate. The enzymes are working at their maximum capacity. This relationship is often described by the Michaelis-Menten curve, a gentle rise that levels off at a maximum rate. It reminds us that enzymes, though efficient, are not limitless. They can only process so many molecules at once, and their speed depends on how easily the substrate binds to the active site. Some enzymes have a strong affinity for their substrate, binding it readily even at low concentrations. Others require higher levels of substrate to achieve the same activity. Beyond substrate concentration, enzymes are also influenced by the presence of inhibitors and activators. Inhibitors are molecules that reduce enzyme activity, either by blocking the active site directly or by binding elsewhere on the enzyme and altering its shape. Some inhibitors are natural regulators, part of the feedback loops that keep metabolism balanced. Others are toxins or poisons, disrupting life by silencing critical enzymes. Activators, by contrast, enhance enzyme activity. They may stabilize the active form of the enzyme, making it more efficient, or they may provide essential cofactors, such as metal ions or vitamins. Without these helpers, many enzymes would remain dormant. For example, the vitamin niacin is required to build NAD, a coenzyme that carries electrons in respiration. Without niacin, the enzymes of energy metabolism cannot function properly. Another subtle factor is the presence of other molecules that compete for the same enzyme. Sometimes two different substrates can bind to a similar active site, leading to competition. In such cases, the relative concentration of each molecule determines the outcome. This competition is another way in which enzymes are regulated, ensuring that pathways adjust depending on what materials are available. Even the cellular environment itself influences enzymes. Crowded with proteins, nucleic acids, membranes, and organelles, the cell is not an empty test tube. The local environment, ionic strength. In some cases, enzymes form complexes with one another, passing substrates directly from one active site to the next. This clustering enhances efficiency, and it too depends on the delicate conditions of the cell. In the natural world, these factors combine in endlessly varied ways. In the soil, enzymes break down organic matter under fluctuating temperatures and pH. In the ocean, enzymes operate in salt-rich waters. In our own bodies, enzymes work in compartments as diverse as the acidic stomach and the neutral blood. Each environment shapes the enzyme, and each enzyme reflects the conditions of its home. As you rest tonight, your enzymes are responding to these factors quietly. They are tuned to the warmth of your body, stable at the pH of your blood, and responsive to the levels of ATP, glucose, and countless other molecules flowing within you. If conditions shift, they adjust, slowing or quickening, always striving to maintain balance. They are both powerful and fragile, resilient yet sensitive, embodying the balance of life itself. Thus, when we speak of the factors that influence enzyme activity, we are reminded of the delicate harmony of existence. Life is possible only because conditions remain within narrow bounds, because enzymes are sheltered within environments that suit them. And while this may sound fragile, it is also steady. Life has persisted for billions of years, through countless changes, always finding ways to adapt its enzymes to new worlds. 9. Enzyme regulation. In the flowing networks of metabolism, enzymes carry out their work tirelessly. They guide each reaction, lower each barrier, and weave molecules into the fabric of life. But life is not simply a matter of reactions occurring at full speed, all the time. If every enzyme in every pathway worked without restraint, the cell would be overwhelmed with activity, resources would be wasted, and balance would be lost. What makes life possible is not just the existence of enzymes, but the ability to regulate them, slowing them down, speeding them up, or pausing them entirely, according to the needs of the moment. This regulation is subtle, continuous, and precise. Just as a symphony requires not only instruments but also a conductor to guide the tempo, enzymes require regulation to ensure harmony. The cell is that conductor, orchestrating millions of reactions with perfect timing, adjusting each one so that the whole remains in balance. One of the most important forms of regulation is feedback inhibition. This is a system in which the product of a pathway turns back to control the very enzymes that began it. Imagine a factory that produces a useful substance. If too much is made, storage overflows and resources are wasted. To prevent this, sensors detect when supplies are full, and the factory slows down production. In the cell, the product of a pathway acts as the sensor. When it accumulates, it binds to an early enzyme in the sequence, reducing its activity. Production then slows or stops, preventing waste. A clear example of this occurs in the synthesis of amino acids. When the amino acid isoleucine is abundant, it binds to the first enzyme in its own pathway, threonine deaminase, and inhibits it. This simple feedback ensures that isoleucine is only produced when needed. When supplies run low, the inhibition is lifted, and the pathway resumes. In this way, the cell avoids both shortage and excess, maintaining balance with elegance. Another form of regulation is allosteric control. The word allosteric means other site. Many enzymes have sites beyond their active site, special regions where molecules can bind and change the enzyme's shape. When a molecule binds to an allosteric site, the enzyme may become more active or less active, depending on the nature of the regulator. This allows fine-tuned control, like adjusting the volume of a sound rather than simply switching it on or off. Allosteric control is often used at critical branch points in metabolism. For instance, phosphofructokinase, a key enzyme in glycolysis, is allosterically inhibited by high levels of ATP. This makes sense. If energy is abundant, there is no need to break down more glucose. On the other hand, when ATP levels fall and ADP or AMP levels rise, phosphofructokinase is activated and glycolysis accelerates. In this way, the cell listens to its own energy state and adjusts its enzymes accordingly. Enzyme regulation also occurs through covalent modification, the reversible addition or removal of chemical groups. One of the most common modifications is phosphorylation, the attachment of a phosphate group. This small change can dramatically alter an enzyme's shape and activity, switching it on or off. Specialized enzymes called kinases add phosphates, while phosphatases remove them. Through these modifications, entire networks of enzymes can be rapidly controlled, responding to signals from inside or outside the cell. In humans and other animals, hormones often act through such modifications. The hormone adrenaline, for example, triggers phosphorylation cascades that activate enzymes involved in breaking down glycogen, releasing glucose for energy. In this way, a signal from the outside world, a need for sudden movement or alertness, is translated into biochemical action through enzyme regulation. Some enzymes require small helper molecules to function, and regulation can occur by controlling the availability of these cofactors or coenzymes. Without magnesium, for instance, certain enzymes cannot catalyze their reactions. Without vitamins such as niacin or riboflavin, enzymes involved in respiration falter. The cell carefully manages these cofactors, ensuring that enzymes receive them only when needed and in the right amounts. There is also regulation at the level of enzyme production. The cell does not always keep large amounts of every enzyme on hand. Instead, it can control which enzymes are made and how much of them are produced by turning genes on or off. When a certain nutrient appears in the environment, the cell may begin producing the enzymes needed to process it. When the nutrient is absent, the enzymes are no longer produced, saving energy and resources. This genetic regulation is slower than direct enzyme inhibition, but it provides long-term control. Another remarkable form of regulation is compartmentalization. In eukaryotic cells, enzymes are confined to specific organelles. This separation allows pathways to occur without interfering with one another. For example, enzymes of respiration are housed in mitochondria, while enzymes of photosynthesis reside in chloroplasts. By controlling where enzymes are located, the cell can regulate when and how they function, releasing them only when the conditions are right. Even the assembly of enzymes into complexes can be a form of regulation. In some pathways, enzymes cluster together, handing substrates directly from one to the next. This increases efficiency and prevents unwanted side reactions. At times, these complexes can be disassembled, reducing activity, and later reassembled when needed. The architecture of enzymes within the cell is therefore itself a tool of regulation. In the broader scale of life, regulation ensures cooperation between tissues and organs. In humans, for instance, enzymes in the liver respond to hormones that reflect the needs of the whole body, storing glucose when food is plentiful and releasing it when energy is required elsewhere. Muscles, in turn, regulate their enzymes differently, favoring rapid ATP production during activity. Each tissue has its own regulatory patterns, yet all are integrated into the balance of the organism. What is most striking about enzyme regulation is its subtlety. It is not brute force, but gentle adjustment, sliding the tempo up or down, opening or narrowing the flow. It allows life to be responsive, adapting to changes in the environment, yet always conserving resources. Through regulation, enzymes do not merely drive reactions. They shape them into patterns that sustain the rhythm of life. As you rest, this regulation is at work within you. If your energy levels fall, enzymes in your cells accelerate the breakdown of glucose. If energy is abundant, they slow down, storing resources for later. Hormones circulate through your blood, carrying messages that adjust enzyme activity across your body. Tiny molecules bind and release, switching enzymes on and off, guiding metabolism with quiet precision. Thus, enzyme regulation is not an exception but the rule. It is the way life maintains its balance, the way cells conserve, adapt, and survive. Without it, enzymes would be wild and uncontrolled, and life would collapse into chaos. With it, enzymes become instruments in a grand orchestra, each playing at the right moment, each contributing to the harmony of living systems. 10. Photosynthesis and energy capture. So far, we have spoken much about how organisms release energy, how glucose is broken down, how electrons flow, how ATP is generated to power life. But energy must first be captured before it can be released. At the foundation of nearly all life on Earth lies one extraordinary process: photosynthesis. It is through photosynthesis that plants, algae, and certain bacteria capture sunlight and convert it into chemical energy, storing it in molecules that can be used by themselves and by every creature that depends on them. In its essence, photosynthesis is the quiet transformation of light into life. Imagine the surface of a leaf, spread wide beneath the sun. To our eyes, the leaf is green, soft, and still. But within its cells, countless reactions are unfolding, guided by enzymes, powered by light, and carried out in perfect harmony. Each photon of sunlight that falls upon the leaf may set in motion a cascade of events that leads to the creation of sugar, a simple molecule that carries within it the energy of the sun. The site of this transformation is the chloroplast, a specialized organelle within plant cells. Chloroplasts contain internal membranes called thylakoids, stacked into structures known as grana. These thylakoid membranes hold pigments, proteins, and enzymes arranged in complexes that capture light and channel its energy into chemical form. The most important pigment is chlorophyll, the green molecule that absorbs light most strongly in the red and blue regions of the spectrum. It is chlorophyll that gives leaves their color, and it is chlorophyll that begins the work of photosynthesis. When a photon of light strikes a chlorophyll molecule, it excites one of its electrons, raising it to a higher energy level. This excited electron does not remain in place. It is passed along a chain of proteins, a journey known as the light reactions of photosynthesis. As the electron moves, its energy is harnessed to pump protons across the thylakoid membrane, building a gradient much like the one we saw in cellular respiration. Eventually, the electron reaches a final acceptor, and along the way, ATP is produced. At the same time, another important molecule is formed, NADPH, a carrier that stores electrons in a high-energy form. Together, ATP and NADPH are the immediate products of the light reactions, serving as the currency of captured sunlight. But where does the electron chain begin? To replace the excited electrons that chlorophyll loses, water molecules are split. This splitting of water releases oxygen, which diffuses out of the leaf into the air. The oxygen we breathe each moment is thus a gift of photosynthesis, the quiet byproduct of water molecules being parted to supply electrons for the process. The light reactions provide energy, but not food. The sugars that sustain plants and, indirectly, all animals are created in the second stage of photosynthesis, the Calvin cycle, sometimes called the light-independent reactions. This cycle occurs in the stroma, the fluid surrounding the thylakoids in the chloroplast. Here, enzymes use the ATP and NADPH from the light reactions to fix carbon dioxide, incorporating it into organic molecules. The key enzyme in this process is ribulose 1, 5-bisphosphate carboxylase, oxygenase. It is one of the most abundant proteins on Earth, and for good reason. Rubisco catalyzes the first major step of carbon fixation, attaching carbon dioxide to a 5-carbon sugar, ribulose bisphosphate. From this beginning, a series of enzyme-guided reactions builds 3 carbon molecules that can eventually be assembled into glucose and other sugars. These sugars become the stored energy of the plant, ready to fuel growth, reproduction, or storage as starch. It is remarkable to consider the simplicity and scale of this process. At its heart, photosynthesis is the joining of carbon dioxide from the air with water from the soil, powered by sunlight, to create sugar and oxygen. 6 molecules of carbon dioxide and 6 molecules of water, in the presence of light, produce 1 molecule of glucose and 6 molecules of oxygen. From this single equation flows the energy of ecosystems, the oxygen of the atmosphere, and the foundation of life as we know it. The sugars created by photosynthesis do not remain in the leaf. They travel through veins, reaching roots, stems, flowers, and fruits. They feed the plant itself, and when animals consume plants, the sugars enter new pathways of respiration, releasing the energy once captured by sunlight. In this way, photosynthesis is not just a plant's process, but the very base of food chains, the entry point of energy into the web of life. Different plants have adapted photosynthesis to their environments in subtle ways. Most use the standard Calvin cycle, known as C3 photosynthesis, because the first stable product has 3 carbons. In hot, dry climates, some plants have evolved C4 photosynthesis, which uses an additional step to capture carbon more efficiently and reduce water loss. Others, such as cacti, use CAM photosynthesis, opening their stomata at night to take in carbon dioxide, conserving water during the day. Each variation reflects the same principle, capturing light, fixing carbon but tuned to different rhythms of the earth. Even in the oceans, photosynthesis thrives. Tiny phytoplankton, drifting near the surface, capture sunlight and fix carbon, producing much of the oxygen in the atmosphere. In forests, towering trees spread vast canopies to catch the sun, while beneath them, mosses, ferns, and smaller plants adapt to dappled light. Across the globe, photosynthesis hums quietly, transforming energy in ways both constant and invisible. It is humbling to realize that every breath we take, every meal we eat, every movement we make, is linked to this process. The oxygen filling our lungs began as water in a plant cell, split by light. The glucose fueling our cells began as carbon dioxide in the air, fixed by Rubisco. Even in darkness, as you listen now, your body is powered by the sun, carried within the bonds of molecules made by photosynthesis. Enzymes, of course, are at the heart of it all. They are the ones that split water, that move electrons, that pump protons, that fix carbon. Without them, the energy of sunlight would remain inaccessible, and life would be impossible. With them, the earth blooms with forests, oceans teem with life, and the atmosphere carries the oxygen that allows us to dream and to rest. Thus, photosynthesis is not only a scientific process, but also a reminder of connection. Light falls upon the earth, and through enzymes, it becomes food, breath, and life itself. It is a continuous cycle. Plants capturing energy, animals releasing it, ecosystems exchanging it, the planet balanced in quiet rhythms. 11. Respiration and energy release. In the last part, we followed the gentle flow of sunlight into leaves, where enzymes captured its energy and stored it in the form of sugars. Now we turn to the other half of this great cycle, the release of that stored energy through respiration. If photosynthesis is the act of filling a vessel with light, respiration is the act of pouring from that vessel, carefully and steadily, so that the energy may be used by every cell. Respiration, in its essence, is the controlled breakdown of glucose and other molecules to release the energy stored in their bonds. Unlike burning, which releases energy all at once as heat and light, respiration proceeds step by step, each reaction guided by enzymes, each step releasing just enough energy to be captured in ATP. It is this measured pace that makes life possible. Were glucose to combust suddenly inside a cell, the result would be chaos and destruction. Instead, enzymes ensure that the energy flows in a steady stream, like a candle glowing gently, rather than a spark exploding in flame. The process begins with glycolysis in the watery cytoplasm of the cell. Here, one molecule of glucose, containing 6 carbons, is split into 2 molecules of pyruvate, each with 3 carbons. Along the way, a small amount of ATP is formed directly, and electrons are transferred to a carrier molecule called NAD, becoming NADH. This first step requires no oxygen and can occur in nearly all living cells, from the simplest bacteria to the most complex animals. For organisms with oxygen, the journey continues into the mitochondria, the organelles often called the powerhouses of the cell. Pyruvate is transported across the mitochondrial membranes and converted into acetyl-CoA, a 2-carbon molecule that enters the citric acid cycle, also known as the Krebs cycle. This cycle, a circular series of enzyme-guided reactions, releases carbon dioxide from the carbons of pyruvate, while transferring electrons to NADH and FADH, another carrier. A small amount of ATP is produced directly, but the main purpose of the cycle is to gather high-energy electrons, storing them in these carriers for the final stage. That final stage is the electron transport chain, located in the inner membrane of the mitochondrion. Here, NADH and FADH deliver their electrons to a series of protein complexes, each one passing the electrons down the line like a relay. As the electrons flow, energy is released, and this energy is used to pump protons across the membrane, building up a gradient. Imagine water being lifted behind a dam. This gradient is a store of potential energy, waiting to be released. The protons then flow back across the membrane through a remarkable enzyme called ATP synthase. Like a molecular turbine, ATP synthase harnesses the movement of protons to attach phosphate groups to ADP, creating ATP. In this way, the cell captures the energy of electrons and translates it into the universal currency of ATP. At the end of the chain, oxygen serves as the final electron acceptor, combining with protons and electrons to form water. This quiet act of receiving electrons is why oxygen is so essential to life, and why each breath we take is, at the molecular level, a participation in respiration. From one molecule of glucose, respiration can yield up to 30 or more molecules of ATP, a remarkable amplification. Each ATP is then used immediately for the countless tasks of the cell, powering enzymes, moving molecules, contracting muscles, firing neurons. Respiration is thus the steady heartbeat of the cell, supplying energy moment by moment, as long as oxygen and nutrients are present. Yet not all respiration requires oxygen. In environments where oxygen is absent, some organisms rely on anaerobic respiration. Here, other molecules, such as nitrate or sulfate, serve as final electron acceptors. In animals, when oxygen is scarce, cells turn to fermentation, converting pyruvate into lactic acid or ethanol, depending on the species. Though fermentation produces far less ATP than aerobic respiration, it is often enough to keep cells alive in difficult conditions. Mussels, for example, use fermentation during intense activity, when oxygen delivery cannot keep up with demand. The result is the temporary buildup of lactic acid, which is later cleared when oxygen becomes available again. Respiration is not limited to glucose alone. Enzymes can channel fats and proteins into the same pathways. Fatty acids are broken down into 2 carbon fragments that feed directly into the citric acid cycle. Amino acids, after being stripped of their nitrogen, can also be converted into intermediates of glycolysis or the cycle. In this way, respiration is flexible, drawing upon many sources of fuel to maintain the flow of energy. What makes respiration so extraordinary is not only its efficiency but also its universality. From the tiniest bacterium to the largest whale, from plants in the forest to fungi in the soil, respiration unfolds with the same basic principles. The molecules may vary, the enzymes may differ, but the logic remains. Step by step, energy is released from food, captured in ATP, and spent on the work of life. We might imagine respiration as the quiet counterpart to photosynthesis. Plants capture light and store it in sugar. Animals, fungi, and many bacteria consume that sugar and release its energy. Even plants themselves use respiration, breaking down some of their own sugars to fuel growth and maintenance. The 2 processes are bound together in a cycle. Photosynthesis fills, respiration empties, and together they sustain the flow of energy through ecosystems. As you rest, respiration continues within you without pause. Each breath brings oxygen into your lungs, which is carried by hemoglobin in your blood to every cell. There, mitochondria receive it, using it to accept electrons at the end of the chain. Each exhale carries away carbon dioxide, the quiet trace of glucose carbons passing through your citric acid cycle. The rhythm of breathing is therefore not just a surface act, but a reflection of deep chemistry, linking your body to the invisible work of enzymes inside your cells. It is humbling to consider that every heartbeat, every thought, every dream relies on this process. Respiration is not dramatic or sudden, but steady and constant. It is the gentle release of energy, always renewed, always balanced, always present, as long as life continues. 12. Special cases. Extremophile enzymes. Until now, we have considered enzymes as they exist in familiar conditions. The warmth of the human body, the neutral pH of blood, the mild environments where most life thrives. Yet, the earth is filled with places that seem hostile to life: boiling hot springs, icy polar oceans, salty desert lakes, and acidic volcanic pools. In these places, conditions appear so harsh that proteins, including enzymes, would be expected to unravel or stop functioning. And yet, life exists there. The organisms that live in such places are called extremophiles, and their enzymes are marvels of adaptation, quietly reshaping our understanding of what is possible for life. Let us begin with thermophiles, organisms that live in high temperatures, often in hot springs or near hydrothermal vents on the ocean floor. For most enzymes, heat is destructive. As temperature rises, proteins unfold, their delicate bonds breaking apart. But enzymes in thermophiles are built to endure. Their amino acid sequences form stronger interactions, often with more ionic bonds and more compact folds, giving them stability even at temperatures that would destroy ordinary proteins. One famous example is Taq polymerase, an enzyme from the bacterium Thermus aquaticus, which lives in Yellowstone's hot springs. This enzyme remains active even at 70 to 80 degrees Celsius, far above the limit for human enzymes. Because of its resilience, Taq polymerase has become a cornerstone of molecular biology, used in laboratories around the world in the process of PCR, or polymerase chain reaction, which copies DNA. What was once a curiosity of hot spring bacteria has become a tool that transformed science. At the other extreme are psychrophiles, organisms that thrive in icy waters, glaciers, and polar seas. For them, the challenge is not heat, but cold. At low temperatures, molecules move slowly, and reactions proceed at a sluggish pace. Enzymes here must remain flexible, able to grasp substrates and guide reactions even when motion is reduced. Psychrophile enzymes often have fewer stabilizing bonds, giving them greater mobility, though this also makes them fragile at higher temperatures. They are finely tuned for the cold, and only the cold. In the vast southern ocean around Antarctica, psychrophilic microbes play an essential role in breaking down organic matter, keeping the cycles of carbon and nutrients moving, even in waters near freezing. Without their enzymes, these cold ecosystems would fall silent. Thus, even in the chill of polar seas, enzymes hum quietly, ensuring life's continuity. Other extremophiles live in conditions of extreme salinity. These are the halophiles, organisms found in places like the Dead Sea or salt flats, where salt concentrations can be 10 times higher than in the ocean. Most enzymes would be destabilized in such briny surroundings, their structures disrupted by the imbalance of ions. But halophiles have enzymes with surfaces adapted to bind water tightly, preventing dehydration, and to tolerate high levels of salt. Their entire biochemistry is adjusted to an environment that would desiccate most cells. Then there are acidophiles, living in acidic environments, such as volcanic hot springs or mines rich in sulfur. The pH in these places can be as low as 1, similar to the strength of stomach acid. For most enzymes, such acidity would denature their structure, disrupting bonds and altering charges. But acidophilic enzymes are stabilized against proton interference, often by shielding sensitive regions or altering amino acid composition. In contrast, alkaliphiles live in highly basic conditions, such as soda lakes, where the pH may rise to 10 or above. Their enzymes, too, are adapted to hold their shape and function despite such unusual chemistry. Another remarkable group are the barophiles, or piezophiles, organisms that live under immense pressure in the deep ocean. At depths of thousands of meters, the weight of water exerts crushing forces. Most enzymes would be distorted, their structures compressed beyond function. But enzymes of barophiles remain flexible under pressure, maintaining activity where we would expect none. Some of these organisms also endure both high pressure and high temperature, dwelling near hydrothermal vents where scalding fluids pour from the Earth's crust. Taken together, these adaptations reveal the resilience and creativity of life. Enzymes, which we once thought fragile, can in fact be shaped to thrive under extremes of heat, cold, pressure, acidity, and salinity. Each extremophile shows that the boundaries of life are wider than we imagined, and each adaptation demonstrates the quiet flexibility of enzymes. These discoveries are not only fascinating in themselves, but also important for science and industry. Enzymes from thermophiles are used in biotechnology, where high temperatures are common. Enzymes from psychrophiles are used in detergents designed to clean at low temperatures, saving energy. Halophile enzymes are studied for their stability in industrial processes involving high salt concentrations. Extremophiles, though living in remote and harsh environments, have provided us with tools that echo through medicine, agriculture, and technology. On a deeper level, extremophiles expand our sense of possibility. If enzymes can adapt to boiling vents, freezing seas, or acidic pools, might life exist in similar environments beyond Earth? The icy oceans beneath the surface of Jupiter's moon, Europa, or the methane-rich lakes of Saturn's moon, Titan, may seem inhospitable, but extremophiles remind us that life finds a way in places we once thought impossible. Their enzymes are testaments to adaptability, whispers of resilience that reach even into the stars. And so, as you rest tonight, consider how diverse and flexible enzymes can be in your body, but elsewhere on Earth, in places of fire, ice, salt, and stone. Enzymes are at work under entirely different rules, yet always fulfilling the same purpose: to guide reactions, to sustain metabolism, to keep life flowing. It is a humbling thought that the same principles which sustain you in comfort also sustain unseen organisms in the most extreme environments on our planet. Life, in all its quiet persistence, depends on enzymes, and enzymes, in turn, show us the extraordinary adaptability of nature. 13. Enzymes in everyday life. So far, we have traveled through the hidden world of enzymes in cells, in pathways, and even in the extremes of the Earth. Yet enzymes are not only deep inside the body or confined to distant, unusual environments. They are present in our daily lives as well. quietly shaping experiences so familiar that we may not even notice them. In this part, we will bring the focus gently closer, exploring enzymes in everyday life, how they help us eat, digest, clean, and even create. The most intimate encounter we have with enzymes happens during digestion. Each time we take a bite of food, enzymes begin their work almost instantly. In the mouth, the enzyme amylase is secreted in saliva. Amylase breaks down starch, a complex carbohydrate found in bread, rice, and potatoes, into simpler sugars. This process is so subtle that you may have noticed it without realizing. If you hold a piece of bread in your mouth for long enough, it begins to taste slightly sweet, as starch is converted to sugar by enzymes. As food travels down into the stomach, other enzymes continue the process. Here, in the acidic environment of gastric juices, pepsin begins to break proteins into smaller fragments. Pepsin works best at a low pH, thriving in the stomach's acidic conditions, where many other enzymes would quickly denature. This careful tuning allows digestion to proceed efficiently. From the stomach, food moves into the small intestine, where an array of enzymes finishes the task. Lipases break down fats into fatty acids and glycerol. Proteases continue the digestion of proteins into amino acids, and amylases and other enzymes complete the breakdown of carbohydrates. By the time nutrients are absorbed into the bloodstream, they are in simple forms: glucose, amino acids, fatty acids that the body can use to build, repair, and generate energy. Without enzymes, the food we eat would remain locked in its complex forms, inaccessible to our cells. Enzymes also play a role in food preparation itself. For centuries, humans have harnessed enzymes in processes such as fermentation. In baking bread, yeast produces enzymes that convert sugars into carbon dioxide and alcohol. The carbon dioxide creates bubbles, causing the dough to rise, while the alcohol evaporates during baking. In brewing beer or fermenting wine, enzymes in yeast break down sugars, producing alcohol that remains. In cheese making, the enzyme renin, or chymosin, helps curdle milk by breaking specific bonds encasing proteins. These ancient practices show that long before we understood enzymes scientifically, we were already working with them, relying on their unseen abilities to transform food. Another everyday encounter with enzymes occurs in laundry detergents. Many modern detergents contain added enzymes that break down stains. Proteases target protein-based stains such as blood or grass. Lipases help remove oily or greasy stains. Amylases break down carbohydrate residues like sauces or chocolate. These enzymes allow stains to be removed at lower washing temperatures, saving energy and making cleaning more effective. It is a quiet example of how enzymes, borrowed from nature, now work alongside us in our households. Enzymes are also present in medicine. In hospitals, certain enzyme-based treatments are used to dissolve blood clots. Enzymes known as fibrinolytics break down fibrin, the protein mesh that holds a clot together, restoring blood flow to tissues. In another case, enzymes are used in diagnostic tests. The familiar blood glucose test, used by people with diabetes, relies on the enzyme glucose oxidase, which reacts with glucose in the sample and helps measure its concentration. In this way, enzymes not only keep us alive but also guide the tools we use to monitor and restore health. Even in agriculture, enzymes are important. In animal feed, enzymes are added to help livestock digest nutrients more efficiently, reducing waste and improving nutrition. In crop protection, Enzyme-based products are being developed to break down pests or pathogens naturally, reducing reliance on harsh chemicals. These applications show how enzymes bridge the natural and the practical, linking biology to daily human needs. In the kitchen, too, enzymes are quietly at work. Consider the ripening of fruit. As a banana ripens, enzymes break down starch into sugars, making it sweet. Other enzymes soften the texture by breaking down cell wall components, and still others change the color and aroma. The transformation of a green banana into a yellow, fragrant, sweet fruit is guided almost entirely by enzymes, acting in gentle coordination. When we slice an apple and watch it slowly turn brown, enzymes are again at play. Polyphenol oxidase, exposed to air, reacts with oxygen and compounds in the apple, producing the brown pigments we see. Though we often think of browning as undesirable, it is simply an enzyme-catalyzed reaction, one that plays a natural role in protecting plants from damage. Enzymes even touch the fibers we wear. In the textile industry, enzymes are used to soften fabrics, remove impurities, and create smooth finishes. Cellulases, for example, break down small fibers on the surface of cotton, making clothes softer and colors brighter. What was once done with harsh chemicals can now be accomplished with enzymes, reducing environmental impact and creating gentler processes. Perhaps one of the most remarkable aspects of enzymes in daily life is their invisibility. We rarely see them or think about them. They leave no mark of their presence except in the results. Food digested, bread risen, stains removed, fruit ripened, clothing softened. Enzymes are subtle workers, their actions hidden, their presence often unnoticed. And yet, without them, so much of daily life would be unrecognizable. As you rest tonight, enzymes are continuing their quiet service within you. They are breaking down nutrients from your last meal, repairing DNA in your cells, clearing away toxins, and sustaining the flow of energy. At the same time, enzymes outside of you are active in bread dough rising in kitchens, in detergents cleansing fabric, in fruits ripening in orchards, and in the soil beneath trees, recycling matter back into the earth. Everywhere, enzymes are present inside and outside, in nature and in human life. They are the silent helpers, linking the invisible chemistry of molecules to the visible rhythm of daily living. And perhaps it is fitting that we rarely notice them, for enzymes do not call attention to themselves. They simply act, continuously and without pause, weaving life together moment by moment. 14. The interplay of enzymes and evolution. Enzymes are not only the workers of life's chemistry in the present moment, they are also ancient storytellers, carrying within their structures the memory of evolution. To understand enzymes is to glimpse the long journey of life on Earth, stretching back billions of years. Each fold of protein, each arrangement of atoms in an active site, has been shaped by time, by change, and by the quiet hand of natural selection. Life began in a world very different from our own. The young Earth was harsh, with no oxygen in its atmosphere, and with oceans filled with simple molecules. The earliest life forms relied on primitive enzymes, proteins, and perhaps even ribozymes, RNA molecules with catalytic power to guide basic reactions. These early catalysts were not as efficient as modern enzymes, but they were enough to sustain metabolism in its simplest forms. The capture of energy from the environment and the assembly of molecules into structures of life. As life diversified, enzymes diversified with it. Each new niche, each new way of living, required changes in metabolism. Enzymes evolved to meet those needs, adapting their shapes, refining their activity, and creating new pathways. Over immense spans of time, enzymes became more specialized, more efficient, and more finely tuned. In this way, enzymes are not only products of evolution, they are also drivers of it, enabling organisms to exploit new sources of energy, new environments, and new ways of living. A striking example of this interplay is the evolution of photosynthesis. Early forms of photosynthesis did not produce oxygen. Instead, they used molecules such as hydrogen sulfide to provide electrons. But eventually, a group of organisms evolved enzymes capable of splitting water, releasing oxygen as a byproduct. This was a turning point in Earth's history, known as the Great Oxidation Event. Around 2.4 billion years ago, the oxygen released by photosynthetic enzymes changed the atmosphere, reshaped ecosystems, and made possible the evolution of aerobic respiration. Thus, enzymes not only evolved but transformed the entire planet. Respiration, too, shows the hand of enzyme evolution. Once oxygen became abundant, organisms with enzymes able to use it for energy production gained a tremendous advantage. The electron transport chains of modern mitochondria are the result of long evolutionary tinkering proteins gradually modified to carry electrons more efficiently. ATP synthase perfected to capture energy from proton gradients. These enzymes enabled life to grow larger, more complex, and more active. Without their evolution, animals, plants, and perhaps even multicellular organisms would never have arisen. Evolution also explains the incredible diversity of enzymes. A single enzyme family can branch into many forms, each adapted for a different role. Consider the proteases, enzymes that break down proteins. Across evolution, proteases have multiplied into dozens of types, each with its own specificity. Some digest food, some remodel tissues, some regulate signaling pathways. All share a common ancestry, yet their functions have diverged to meet the varied needs of organisms. The process behind this diversity is often gene duplication. When a gene for an enzyme is copied, one version can maintain its original function, while the other is free to mutate and evolve. Over time, the second version may develop a new specificity or work under different conditions. This mechanism has allowed enzymes to expand their roles, creating the vast catalog of catalysts we see today. Another dimension of enzyme evolution is adaptation to environments. Extremophiles, which we considered earlier, are excellent examples. Their enzymes bear the marks of evolutionary adjustment to heat, cold, acidity, or pressure. Each change in amino acid sequence is a record of natural selection, a fine-tuning that allows the enzyme to function in places where life might otherwise fail. In this way, enzyme evolution tells the story of life's persistence across the most unlikely landscapes. Interestingly, not all enzymes are perfect. Some are slower or less efficient than they might be. This is because evolution does not strive for perfection, but for sufficiency. If an enzyme works well enough to sustain survival and reproduction, it may not evolve further. The enzyme rubisco, central to photosynthesis, is a classic example. It is both ancient and inefficient, often binding oxygen instead of carbon dioxide, leading to wasteful reactions. And yet, because it performs adequately in most conditions, it remains one of the most abundant enzymes on Earth. Rubisco's imperfection is itself a lesson in evolution's balance between efficiency and adequacy. Enzymes also reveal connections between species. When scientists compare enzyme sequences from different organisms, they can trace evolutionary relationships, seeing where lineages diverged and how proteins have changed. In this way, enzymes serve as molecular fossils, preserving the history of life within their structures. For instance, enzymes in humans often show close similarity to those in other animals, reflecting shared ancestry. Even in plants, fungi, and bacteria, echoes of common enzymatic origins can be found. Beyond Earth, the story of enzyme evolution invites us to wonder: if life exists elsewhere, it too would require catalysts to guide its chemistry. They might not be proteins as we know them, but they would play a similar role, lowering barriers, sustaining reactions, weaving energy into the fabric of living systems. In imagining life on distant worlds, we are, in a sense, imagining what other forms of enzymes might have evolved. And so, enzymes are both products and partners of evolution. They have been shaped by billions of years of change, yet they have also driven those changes, making new forms of life possible. From the first primitive catalysts to the complex enzymes within us, they trace the arc of life's history. As you drift into rest, consider that the enzymes in your body carry within them echoes of ancient seas, of microbes that once lived in the deep past, of adaptations that allowed life to flourish in countless forms. They are timeless companions, born of evolution, sustaining the present and carrying life forward into the future. 15. Energy balance and homeostasis. Life is not only about the capture and release of energy, it is about balance. Within every organism, from the simplest bacterium to the human body, conditions must be kept within narrow ranges. Too much energy or too little, too many nutrients or too few, too high a temperature or too low, and the harmony of life begins to falter. The word for this balance is homeostasis, a quiet state of stability that allows cells and organisms to function smoothly. At the heart of this balance are enzymes, continuously adjusting the flow of energy so that life remains steady and secure. To understand homeostasis, we might imagine the body as a finely tuned orchestra. Each instrument represents a process: digestion, respiration, circulation, temperature regulation. If one instrument plays too loudly or too softly, the harmony is disturbed. Enzymes act as the quiet conductors, keeping each process in rhythm, responding to changes, and ensuring that no single part overwhelms the whole. A clear example of this balance is seen in blood glucose regulation. Glucose is the primary fuel for many cells, especially in the brain, but too much glucose in the blood can damage tissues, while too little leaves cells starved of energy. Enzymes are central to maintaining this delicate range. After a meal, enzymes in the liver convert excess glucose into glycogen, a storage form. When fasting or exercising, enzymes break down glycogen back into glucose, releasing it into the blood. Hormones like insulin and glucagon direct this process, but the actual work is carried out by enzymes, responding to signals and adjusting their activity. This regulation extends to ATP itself, the cell's direct energy currency. Levels of ATP, ADP, and AMP act as signals of energy status. When ATP is abundant, enzymes slow down pathways that produce more, preventing waste. When ATP falls and AMP rises, enzymes activate pathways that generate energy, such as glycolysis. Through feedback and allosteric control, enzymes sense the cell's needs and shift their activity accordingly. In this way, the cell avoids both energy shortage and excess, keeping itself in steady equilibrium. Enzymes also contribute to temperature balance. Although the body uses physical mechanisms such as sweating and shivering, Enzymes are the reason why temperature must remain within narrow limits. Human enzymes, for instance, work best near 37 degrees Celsius. If temperature rises too high, they begin to denature. If it falls too low, their activity slows. Thus, the entire system of thermoregulation, warming, cooling, adjusting, is in place to preserve the conditions in which enzymes can function properly. Homeostasis, at its core, is often about maintaining environments suitable for enzymes. Another dimension of homeostasis involves pH regulation. The blood maintains a nearly constant pH around 7.4. If it shifts even slightly, enzyme activity can be disrupted. Buffers in the blood, along with enzymes in the kidneys and lungs, work constantly to hold this pH steady. Carbonic anhydrase, an enzyme in red blood cells, is one example. It rapidly converts carbon dioxide and water into carbonic acid, which can then release or absorb protons, helping maintain pH balance. Without such enzymes, the chemistry of life would quickly drift out of range. Even the disposal of waste products relies on enzymes for balance. Ammonia, a byproduct of protein metabolism, is toxic in high amounts. Enzymes in the liver convert ammonia into urea, which can be safely excreted. Similarly, enzymes neutralize reactive oxygen species, harmful molecules produced during respiration. Catalase, for example, breaks down hydrogen peroxide into water and oxygen, protecting cells from oxidative damage. These actions, though invisible to us, are essential to homeostasis, preventing small imbalances from growing into threats. In larger organisms, enzymes support the integration of multiple systems. For example, during exercise, muscles demand more ATP. Enzymes in muscle cells accelerate glycolysis and respiration. At the same time, enzymes in the liver release glucose into the blood, while enzymes in red blood cells help deliver oxygen. All of these adjustments are coordinated through hormones and signals, but the real effectors are enzymes, quietly shifting their activity to match the body's needs. The result is a seamless supply of energy, rising and falling like a tide, always adapted to the moment. Homeostasis is not static, it is dynamic, a constant adjustment to change. Enzymes are central to this adaptability. When nutrients are plentiful, they guide storage. When nutrients are scarce, they guide mobilization. When oxygen is present, they favor aerobic respiration. When oxygen is absent, they switch to fermentation. These shifts happen smoothly, without conscious thought, driven by the responsiveness of enzymes to their surroundings. In ecosystems, too, homeostasis can be seen, with enzymes as the mediators. In soils, enzymes released by microbes regulate the decomposition of organic matter, recycling nutrients at just the right pace to sustain plants. In oceans, enzymes of phytoplankton balance the uptake of carbon dioxide, influencing global carbon cycles. Even at the scale of the planet, enzymes contribute to equilibrium, their countless small actions adding up to stability in Earth systems. It is remarkable that something as small as an enzyme molecule can play such a large role in balance. Each enzyme acts only on its own substrate, performing one specific reaction, yet together they create a network of checks and adjustments, a system that maintains life's stability in the face of constant change. This is the quiet miracle of homeostasis, that millions of individual reactions, guided by countless enzymes, add up to harmony. As you drift deeper into rest, your enzymes are at work, maintaining this balance within you. They are adjusting your blood sugar, buffering your pH, managing your temperature, and clearing away wastes. They are ensuring that your cells neither hunger nor overflow, that your energy flows steadily, that your body remains in quiet equilibrium. You may not feel their work, but you are sustained by it, every moment, without pause. Thus, enzymes are not only catalysts of reactions, they are guardians of balance. They are the unseen stewards of homeostasis, holding life steady in the delicate ranges where it can flourish. Without them, energy would be wasted, chaos would rise, and stability would collapse. With them, life moves gently forward, poised between constancy and change, always in balance. 16. The fragility and power of enzymes. Enzymes, as we have seen, are the gentle workers of life. They speed reactions, maintain balance, and guide the flow of energy. Yet they are not invincible. Enzymes are both fragile and powerful, delicate in their requirements, yet immense in their effects. This duality is part of their wonder, the way such small and sensitive molecules hold the strength to sustain entire worlds. Let us begin with their fragility. Enzymes are proteins, folded into precise shapes. Their activity depends entirely on the stability of these shapes, but the bonds that hold proteins together are not unbreakable. A rise in temperature can cause them to unravel. A shift in pH can alter charges and disrupt their folds. A lack of necessary cofactors can silence them. Even slight imbalances in the cellular environment can weaken their function. This sensitivity explains why living organisms must maintain homeostasis so carefully. It is not only for the comfort of the organism as a whole, but for the survival of its enzymes. A vivid example of this fragility is seen in fever. When body temperature rises even a few degrees, some enzymes begin to lose their efficiency. If the temperature rises too high, they denature, losing their shape entirely. Similarly, in freezing conditions, enzyme activity slows to a crawl, and life itself enters a kind of suspended state. These changes remind us that enzymes live within narrow ranges, and when those ranges are breached, the foundations of life are shaken. Another example lies in toxins and poisons, many of which act by interfering with enzymes. Cyanide, for instance, binds to an enzyme in the electron transport chain, blocking the flow of electrons and halting respiration. Without that one enzyme functioning, ATP production collapses and cells cannot survive. It is sobering to realize that the fragility of enzymes can be a point of vulnerability, one exploited by nature and, at times, by human misuse. Yet alongside this fragility lies their power. Even though enzymes require such precise conditions, within those conditions they are astonishingly effective. A single enzyme molecule can catalyze thousands of reactions per second. Without enzymes, many reactions in the cell would take years, centuries, or longer to occur on their own. With enzymes, they happen instantly, at body temperature, in the gentle environment of the cell. This power is not only a matter of speed but of direction. Enzymes bring order to chemistry. They channel reactions along specific pathways, avoiding chaos. In the crowded environment of a cell, where countless molecules float and collide, their specificity is immense. Most enzymes act on only one substrate, sometimes even distinguishing between molecules that differ by a single atom. The combination of fragility and power gives enzymes a unique role in life. They are like finely tuned instruments sensitive to the slightest change, yet capable of producing remarkable effects when conditions are right. Their power depends on their delicacy, and their delicacy is balanced by the immense networks in which they operate. Life thrives not by making enzymes indestructible, but by building environments that protect them, and by multiplying their effects through numbers and cooperation. Consider the enzyme DNA polymerase, which copies genetic material. Its task is monumental: to replicate billions of nucleotides in human DNA with remarkable accuracy, so that cells can divide and life can continue. Yet this enzyme is still fragile. It requires the right temperature, the right magnesium ions, the right balance of nucleotides. Without these, it cannot function. But when all is provided, it performs a task that sustains heredity itself. Or consider ATP synthase, the turbine-like enzyme that generates most of the ATP in cells. It is delicate in its structure, dependent on the flow of protons across a membrane. Yet it harnesses that flow with extraordinary power, producing energy that fuels nearly every activity of life. One cannot help but marvel at how something so tiny and vulnerable underpins everything from the beating of a heart to the opening of a flower. This interplay of fragility and power also explains why life is so dependent on stability. Organisms have evolved not to change the nature of enzymes, but to protect the conditions they need. Warm-blooded animals maintain body temperature for their enzymes. Plants regulate the chemistry of their cells to shield enzymes from drought or salinity. Even extremophiles, which appear to defy limits, do so not by making enzymes that ignore conditions altogether, but by evolving enzymes delicately adapted to those specific extremes. There is also beauty in how this balance echoes through ecosystems. The fragility of enzymes means that life as a whole depends on narrow ranges of temperature, pH, and chemistry. The power of enzymes means that, within those ranges, life flourishes abundantly. Together, they weave a pattern of resilience and vulnerability, one that has persisted across billions of years. For us, the lesson may be one of humility. Our lives depend on molecules so small we cannot see them. Molecules that fold into shapes so delicate they can be undone by a degree or two of heat, and yet so potent they sustain every heartbeat, every breath, and every thought. The fragility of enzymes reminds us of the preciousness of balance. Their power reminds us of the strength hidden in the smallest forms. As you rest now, your enzymes continue their quiet work. They are fragile, and so your body shields them, keeping your temperature, pH, and chemistry within safe ranges. They are powerful, and so they transform food into energy, maintain your cells, and sustain your mind even as you sleep. In their delicacy, they depend on balance. In their strength, they give rise to life itself. Thus, enzymes embody both vulnerability and resilience. They are the delicate threads, and yet they are also the anchors of existence. They remind us that power need not come from brute force. It can come from precision, from balance, and from quiet constancy. 17. Enzymes and the future of biotechnology. Throughout our journey, we have seen how enzymes shape the hidden chemistry of life. They digest food, guide energy, maintain balance, and even adapt to extremes. But the story of enzymes does not end within cells or in natural ecosystems. Humans, with growing knowledge, have begun to harness enzymes in deliberate ways, using them not only as helpers in daily life, but also as tools for the future. This unfolding field is known as biotechnology, and enzymes stand at its heart, offering possibilities for medicine, energy, food, and sustainability. Let us begin with medicine. Enzymes already play quiet roles in diagnostics, helping measure glucose, cholesterol, and other markers in blood. But the future holds far more. Scientists are engineering enzymes to target diseases directly. In cancer therapy, for example, certain enzymes are being designed to break down protective barriers around tumors, making treatments more effective. In genetic medicine, enzymes such as CRISPR-associated nucleases can cut DNA with precision, allowing scientists to correct mutations or add new genes. These tools are built on enzymes that bacteria evolved long ago to defend themselves against viruses, now repurposed to heal human bodies. Enzymes are also being developed as medicines themselves. Enzyme replacement therapy is already used for certain rare conditions where patients lack a crucial enzyme. In the future, more enzymes may be delivered directly to patients, breaking down harmful molecules or restoring missing functions. These therapies highlight how the delicate work of enzymes inside the cell can be extended outward, offering hope for conditions once thought untreatable. Beyond medicine, enzymes are finding new roles in sustainable energy. The world faces the challenge of reducing reliance on fossil fuels, and enzymes may help. Certain enzymes can break down plant material, such as cellulose, into sugars that can then be fermented into biofuels. These enzymes are being refined to make the process more efficient, opening the possibility of renewable energy from agricultural waste and non-food crops. Other enzymes are being studied for their ability to split water or capture carbon dioxide, potentially contributing to clean energy and climate solutions. In agriculture, enzymes promise gentler and more efficient practices. Enzymes added to animal feed improve digestion, reducing waste and the need for excess resources. Enzyme-based fertilizers are being explored, designed to release nutrients gradually, reducing runoff and environmental damage. Enzymes may also provide natural defenses against pests, replacing harsh chemicals with precise biological tools. These possibilities point to a future where agriculture is both productive and sustainable, guided not by brute force but by the subtle action of enzymes. Food production, too, is being reshaped by enzymes. Already, enzymes help make bread, cheese, beer, and yogurt. In the future, enzymes may help create plant-based or cultured alternatives to meat and dairy, making them more nutritious and appealing. Enzymes can adjust flavors, textures, and nutritional profiles, helping to craft foods that are healthier and more sustainable. They can also extend shelf life, reducing waste in global food systems. In this way, enzymes may contribute not only to feeding people, but also to caring for the planet. Another frontier lies in bioremediation, the use of enzymes to clean the environment. Some enzymes can break down pollutants such as oil, plastics, or pesticides. Microbes with specialized enzymes are being studied to remove toxins from soil and water. One striking example is the discovery of enzymes that can degrade polyethylene terephthalate, or PET, a common plastic. Engineering these enzymes could help reduce plastic pollution, one of the major environmental challenges of our time. Enzymes, once again, show themselves as both natural tools and future allies. Perhaps most fascinating is the field of enzyme engineering. Using techniques such as directed evolution, scientists can create new enzymes that nature has never produced. In directed evolution, enzymes are made to mutate and adapt in the laboratory, and versions with desired properties are selected. This mimics natural selection but accelerates it, allowing enzymes to be tailored for specific tasks. Some engineered enzymes are more stable, faster, or able to work in unusual conditions. Others perform reactions unknown in nature, expanding the very toolkit of life. This ability to design enzymes is opening possibilities across industries. Enzymes that withstand high heat may be used in industrial processes. Enzymes that work in solvents may aid in chemical synthesis. Enzymes with altered specificities may produce new medicines or materials. What once took millions of years of evolution can now, in some cases, be guided in laboratories over months or years. Even in space exploration, enzymes may have roles. If humans travel to distant worlds, enzymes could help recycle waste, produce food, or generate fuel in closed environments. Extremophile enzymes, already adapted to unusual conditions, may serve as models for life support systems beyond Earth. Enzymes, it seems, are not only part of life's past and present, but may also accompany us into the future, wherever we go. And yet, in thinking of these possibilities, we must remember the nature of enzymes themselves. They are fragile and require balance. To use them wisely, we must create conditions that respect their needs and limits. Biotechnology's future lies not in forcing enzymes into unnatural extremes, but in working with their strengths, guiding their activity gently, just as life has always done. As you listen, perhaps imagine enzymes as tiny keys, each fitting a single lock, each unlocking a possibility. In the future, humanity may craft new keys, open new locks, and discover new pathways. But the principle will remain the same: subtlety, precision, and the quiet power of catalysis. Thus, enzymes are not only ancient molecules of the past, not only present workers of the cell, but also companions in the journey ahead. They connect evolution to innovation, biology to technology, life to sustainability. In their fragility lies care, in their power lies promise, and in their future lies the hope that we may live more gently with the earth, guided by the same catalysts that first made life possible. 18. The harmony of enzymes and energy. As we near the gentle close of this session, it is time to pause and look back at the patterns we have followed. From the first introduction of enzymes to their role in energy capture, release, balance, and even their applications in the future, a common thread has emerged: the harmony of enzymes and energy. Life, in all its forms, is shaped by this relationship. Enzymes guide energy's flow, and energy, in turn, sustains the work of enzymes. Together, they form a cycle, fragile yet enduring, quiet yet immense. Think of energy as the stream that nourishes the landscape of life. Left on its own, it might rush chaotically, carving paths unpredictably, or stagnate, leaving areas untouched. Enzymes are like gentle hands shaping the stream, guiding it into channels, slowing it here, quickening it there, ensuring that the flow nourishes rather than destroys. The balance between energy and enzymes is thus not a contest of strength, but a cooperation, a harmony that allows life to exist in ordered rhythms. In photosynthesis, we saw how light energy is captured by pigments and converted into chemical forms, ATP and NADPH, then woven into sugars by enzymes. In respiration, we saw how sugars are broken down step by step, their energy released and recaptured in ATP. In both cases, enzymes were the mediators, ensuring that the energy was not lost in chaos, but delivered precisely to where it was needed. Energy alone could not create life. Enzymes alone could not sustain it. But together, they create the foundation of existence. This harmony is reflected in homeostasis, the delicate balance within organisms. Enzymes adjust their activity in response to energy status, while energy availability shapes how enzymes are used. If energy rises too high, enzymes slow its production. If energy falls too low, enzymes accelerate pathways to restore it. In this way, enzymes and energy maintain a dialogue, responding to each other's signals, never static, always in motion, like two partners in a dance. On the scale of ecosystems, the same balance can be seen. Plants capture energy, animals release it, decomposers recycle it, and enzymes are present at every step. In soil, enzymes break down fallen leaves into nutrients. In oceans, enzymes within phytoplankton fix carbon. In animals, enzymes in digestion release the energy of food. Each organism plays its part, but the unifying principle is the same. Enzymes guiding energy through cycles, weaving life together in quiet, unseen harmony. The fragility and power of enzymes make this harmony all the more profound. Their fragility means they cannot act in isolation. They require stable conditions, environments that protect them. Their power means that once given those conditions, they can sustain entire systems. Life thus exists not by overcoming fragility, but by embracing it, creating networks, balances, and protections that allow enzymes to do their work. Energy, too, is fragile in its own way, prone to dissipation, to waste, to loss. As heat, enzymes preserve it, capturing it in usable forms, extending its usefulness, turning fleeting flows into enduring cycles. This harmony also extends across time. Enzymes are ancient, shaped by billions of years of evolution. Energy has flowed across the earth since the beginning, from sunlight, from chemical gradients, from the heat of the planet itself. Together, enzymes and energy have coexisted, co-adapted, and co-evolved, giving rise to microbes, to plants, to animals, and finally to us. The continuity of life across ages is, at its foundation, the continuity of enzymes guiding energy. Even in the future, as we look to biotechnology, sustainability, and perhaps even life beyond Earth, the principle will remain the same. Whatever new enzymes we design, whatever new energy sources we harness, the interplay between them will determine success. The harmony of enzymes and energy is not confined to Earth's past or present. It is a universal principle of life itself. As you listen now, your own body is a living example of this harmony. Enzymes in your cells are breaking down glucose, releasing energy in small steps. That energy is being captured in ATP, which powers countless reactions, repairing DNA, sending signals between neurons, maintaining the steady rhythm of your heartbeat. Meanwhile, enzymes are balancing energy use, ensuring that nothing is wasted, that all remains in quiet equilibrium. Every breath you take is part of this harmony, oxygen serving as the final electron acceptor, carbon dioxide released, enzymes guiding each step in between. It is easy to overlook such harmony because it is invisible, constant, and silent. We notice the outcomes, growth, movement, thought, but not the hidden partnership that makes them possible. Yet beneath all the complexity of life, there is a simple truth. Enzymes and energy are inseparable, two aspects of the same flow. Energy provides the possibility, enzymes provide the precision. Together, they sustain the wonder of living systems. And so, as we bring this session to its close, let us reflect softly on this partnership. It is not dramatic, not forceful, not loud. It is gentle, steady, and enduring. It is the quiet hum within leaves, within oceans, within cells, within you. The harmony of enzymes and energy is the harmony of life itself. 19. A gentle recap of energy and enzymes. We have covered much ground in this quiet exploration, moving step by step through the hidden chemistry of enzymes and the flow of energy. Before we reach our soft conclusion, let us pause here for a gentle recap, gathering together the threads of what we have learned, so that the picture rests clearly and calmly in the mind. We began with the simple idea of biochemistry, the study of the molecules that make life possible. Within this vast field, we focused on enzymes, the catalysts of living systems, and on energy, the force that fuels every activity. These two themes have been our companions throughout this journey: enzymes as guides, and energy as the flow they guide. We looked at what energy means in life, not only in the physical sense of movement or heat, but as the chemical potential stored in the bonds of molecules. This energy, captured and released, is the thread that connects every cell, every organism, and every ecosystem. From there, we discovered ATP, the universal energy currency of life. It is a small molecule, but it carries enormous significance. Each phosphate bond it contains is a tiny reservoir of potential, ready to be released when needed. ATP is the language in which energy is spoken across all of life's domains. We then turned to enzymes as biological catalysts, learning how they lower activation energy, making reactions possible at the gentle conditions of life. Without enzymes, chemical changes would be too slow, too uncertain. With enzymes, life unfolds in rhythm, reactions moving smoothly along their pathways. We examined the structure of enzymes, their active sites, and their specific fits with substrates. This precision explained why enzymes are so effective, and why even small changes in their shape can alter or disrupt their function. From there, we explored the pathways of metabolism, glycolysis, the citric acid cycle, oxidative phosphorylation. We saw how energy is extracted step by step, guided by enzymes, so that nothing is wasted and everything remains in balance. Enzymes transformed metabolism from chaos into order, from random reactions into carefully tuned systems. We noticed how enzymes connect pathways into networks, allowing cells to adapt and adjust. Their activity is influenced by temperature, pH, and concentration, showing both their fragility and their responsiveness. Regulation through inhibitors, activators, and feedback loops ensures that enzymes do not act too little or too much, but just enough to sustain equilibrium. We widened our view to photosynthesis and respiration, where enzymes capture sunlight in plants and release stored energy in animals. Here we saw the great circle of energy: light transformed into sugars, sugars broken down into ATP, and ATP fueling the quiet activity of cells. We stepped briefly into extremes, discovering extremophile enzymes, thriving in heat, cold, acidity, pressure, and salt. These showed us the adaptability of life and hinted at the possibility of enzymes in alien worlds. We returned to the familiar, noticing enzymes in everyday life, in digestion, in ripening fruit, in bread rising, in laundry detergents, in medicine. Enzymes, we realized, are not confined to textbooks or distant ecosystems. They touch us daily, often invisibly. Then, we placed enzymes in the grand story of evolution. They are ancient molecules, shaped across billions of years, both products and drivers of change. From primitive catalysts in early oceans to the highly specialized enzymes of today, they tell the history of life in their folds and sequences. We reflected on their role in homeostasis, in the balance that sustains living beings. Enzymes help regulate glucose, ATP, temperature, pH, and waste products. They are the guardians of equilibrium, keeping conditions within the delicate ranges life requires. We then acknowledge their fragility and power, their vulnerability to small changes, and yet their immense capacity to sustain life when conditions are right. Fragile, but indispensable. Delicate, yet powerful. Finally, we glimpse the future of biotechnology, where humans learn to harness enzymes more directly. From medicine to clean energy, from agriculture to environmental care, enzymes promise to be partners in creating more sustainable paths forward. Through all of this, a single theme has persisted: the harmony of enzymes and energy. One provides the flow, the other provides the guidance. Together, they create the rhythm of life, a rhythm that has continued for billions of years and will continue still. And now, having walked this path gently, we are ready to let the story close in quiet reflection. In the final part, we will bring all these threads together: enzymes, energy, balance, and life itself into a soft conclusion, a meditative closing to accompany rest. 20. A quiet closing reflection. We have now reached the final stage of our long and unhurried journey. Together, we have wandered through the hidden landscapes of enzymes and the flow of energy. We have seen how these small, delicate catalysts sustain the grand patterns of life, from the breaking down of a single molecule of sugar to the cycling of oxygen and carbon across the earth. Now, in this last part, we will not introduce new knowledge or explore new systems. Instead, we will rest gently with what we have gathered, weaving it together into a soft reflection. When we think of life, we often imagine visible things: a forest of trees, a body in motion, the movement of animals across a landscape, the changing of seasons. Yet beneath all of these lies a subtler truth. At the smallest scales, invisible to the eye, enzymes are quietly at work. They are not loud, not forceful. They act softly, lowering barriers, guiding reactions, channeling energy. Their work is constant and unending, yet always delicate. Energy, too, is quiet. We do not see it directly, yet we feel its presence everywhere. It is the sunlight warming the surface of the earth. It is the heat in our bodies. It is the strength in our muscles and the thoughts in our minds. Energy flows, but without guidance, it would disperse, dissolve into nothing useful. Enzymes give it shape, directing it into the pathways of life. Together, enzymes and energy form a partnership as old as time, a harmony that sustains every living being. Think of the journey energy takes through a single cell. Glucose enters, a simple sugar molecule. Step by step, enzymes dismantle it, releasing its potential. At each step, the energy is captured in ATP, stored for later use. Other enzymes then take that ATP and deliver it to reactions, contracting a muscle fiber, transmitting a nerve signal, repairing a strand of DNA. Without enzymes, glucose would remain locked, inaccessible. Without energy, enzymes would have no purpose. Only together do they make life possible. The same is true in plants. Sunlight, seemingly intangible, is caught by pigments and passed into enzymes that fix carbon dioxide into sugar. This sugar becomes food for plants themselves, and for animals, and for us. The energy of the sun, millions of kilometers away, is thus guided gently into the flow of life by enzymes, folded proteins working quietly in green leaves. Energy becomes matter, matter becomes energy again, and the cycle continues. This partnership is not confined to individual organisms, it extends into entire ecosystems. In the soil, microbial enzymes decompose fallen leaves, releasing nutrients back into the earth. In rivers and oceans, enzymes within plankton and bacteria recycle the remains of other organisms, keeping cycles moving. Even the atmosphere carries the marks of enzymatic work. The oxygen we breathe is the gift of enzymes in plants and algae, tirelessly splitting water and fixing carbon over billions of years. It is a vast and interconnected rhythm, yet at its foundation, it remains the same as within a single cell. Energy flows, enzymes guide, balance is maintained, life continues. The story of enzymes and energy is also the story of fragility and resilience. Enzymes are delicate, undone by shifts in temperature or pH, reliant on narrow conditions. And yet, when those conditions are provided, they are powerful beyond measure, able to sustain worlds. Energy, too, is fleeting, always seeking to dissipate, always tending toward loss. And yet, when captured and directed, it fuels every heartbeat, every breath, every green leaf turning toward the sun. The strength of life lies not in invincibility, but in this balance of fragility and resilience. Even as you rest now, this balance continues within you. Enzymes in your cells are breaking down nutrients from your last meal. They are repairing DNA, synthesizing proteins, maintaining pH, and clearing wastes. They are keeping your heart beating at its steady rhythm, ensuring that your brain has glucose to fuel its gentle dreams. They do not stop when you close your eyes. They do not pause when you drift into sleep. Their constancy is what makes rest possible. At the same time, energy flows quietly through you. The oxygen from your breaths is carried to cells, received by enzymes in mitochondria, and used to complete respiration. Carbon dioxide, the byproduct, is carried away, exhaled without thought. With each cycle of breathing, energy is released, captured, and renewed. It is a process older than humanity, older than forests, older even than oxygen itself in Earth's atmosphere. It is the same process that began in ancient seas billions of years ago and has continued unbroken to this moment. There is a stillness in recognizing this continuity. The same patterns that sustain you tonight also sustained the first microbes on Earth, the first plants that grew in sunlight, the first animals that walked on land. They will continue tomorrow and the day after, long after you wake again. Enzymes and energy are not temporary. They are the steady hum beneath all change, the quiet rhythm that carries life forward through time. As we conclude, it may help to imagine this harmony as a kind of music, not a loud or forceful melody, but a background hum, steady, patient, eternal. Each enzyme is a note, each reaction a chord, each flow of energy a movement in the composition. Together, they form the music of life. It is soft, constant, and unbroken, surrounding us always. And so, the lesson of enzymes and energy is also a lesson for us. Life is not sustained by force, but by balance, not by noise, but by quiet constancy, not by dramatic power, but by precision, patience, and cooperation. Fragile things can be strong, Small things can sustain great systems. Quiet things can be the foundation of existence. Tonight, as you rest, let that thought carry you gently. Your body is part of this rhythm. Your cells are part of this music. The enzymes will continue their quiet work, and energy will continue its gentle flow. You need do nothing. Balance will hold, constancy will remain, and life will continue, softly, endlessly, in the harmony of enzymes and energy. Rest now, in peace, in stillness, in quiet trust. The rhythm will go on, the enzymes will guide, the energy will flow, and you will be carried by them, safe in the calm of night.